Centrosome movement, spindle formation, and nuclear envelope breakdown occurs as in mitosis. Chromosomes condense progressively throughout prophase 1. During early prophase 1, before the stage shown above, each chromosome pairs with its homologue aligned gene by gene, and crossing over occurs. The DNA molecules of non-sister chromatids are broken by proteins and are rejoined to each other. At the stage shown above, each homologous pair has one or more X-shaped regions called chiasmata, where crossovers have occurred. Later in prophase 1, after the stage shown above, microtubules from one pole or the other will attach to the two kinetochores, one at the centromere of each homologue. The homologous pairs will then move toward the metaphase plate. Pair of homologous chromosomes are now arranged at the metaphase plate, with one chromosome in each pair facing each pole. Both chromatids of one homologue are attached to kinetochore microtubules from one pole. Those of the other homologue are attached to microtubules from the opposite pole. Breakdown of proteins that are responsible for sister chromatid cohesion along chromatid arms allows homologues to separate. The homologues move toward opposite poles guided by the spindle apparatus. Sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit toward the same pole. When telophase 1 begins, each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. One or both chromatids include regions of non-sister chromatid DNA. Cytokinesis usually occurs simultaneously with telophase 1, forming two haploid daughter cells. A single apparatus forms in late prophase 2, chromosomes, each still composed of two chromatids, associated at the centromere, move toward the metaphase 2 plate. The chromosomes are positioned at the metaphase plate as in mitosis. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. Breakdown of proteins holding the sister chromatids together at the centromere allows the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. Nuclei form. The chromosomes begin decondensing and cytokinesis occurs. The meiotic division of one parent cell produces four daughter cells, each with a haploid set of unduplicated chromosomes. The four daughter cells are genetically distinct from one another and from the parent cell. Copying errors may lead to alterations within the sequence of a gene. A change may have no effect on the resulting gene product or may severely disrupt the function of the gene alternations, which disrupt gene function and lead to diseases known as mutations. Meiosis produces haploid gametes that are genetically different. This is due to two processes: independent assortment and crossing over. Essentially, these processes shuffle alleles during gamete formation and create genetic variation.